Hello students, now we will start with new topic, that is trisaccharides. Means the compound is made up of 3 monosaccharides or 3 monomers of carbohydrates. 3 monosaccharides. The compound is made up of 3 monosaccharides. Here we have to go for example, that is raffinose. Raffinose. So raffinose is made up of glucose plus galactose plus fructose. We also have seen the term like oligosaccharides for the carbohydrates that are made up of 3 or more than 3 number of monosaccharide units. So we will see next term, that is oligosaccharide. Even raffinose also can be studied under oligosaccharide. But just for your understanding, I am giving this one oligosaccharide. Means your compound that is made up of 3 or more than 3 number of monosaccharides. Here example we can take as hemicellulose. Hemicellulose. So here hemicellulose, it is made up of nearly 5 types of monosaccharides. Actually hemicellulose also exists in the form of polysaccharide also. Under heteropolysaccharides we read one best example, but since it is having exactly so and so number of, it is made up of 1 unit of hemicellulose molecule is made up of 5 different types of monosaccharides. We are also studying that one here. That is 1 unit of hemicellulose. It is made up of xylulose, mannose, arabinose. We can also call this one xylulose as xylanes also. XYLANS, xylanes. Next, another one, galactose. Another monosaccharide, glucose. Cellulose is a 5-carbon compound that we see in a regeneration of C3 cycle. Mannose also we will read in algae chapter. When we are reading about chlorophyce, pheophyce, rhodophyce, then we come across the word mannose. Mannose is a 6-carbon compound. Arabinose is a 5-carbon compound. Galactose is a 6-carbon compound. Next, glucose is a 6-carbon compound. So 1 unit of hemicellulose is made up of 5 different types of monosaccharides. But when hemicellulose is existed in plant body, then hemicellulose will be existed in the form of polysaccharide. Means many number of hemicellulose units will form into one complete layer. When you are so polysaccharides means they are made up of many monosaccharides. Under polysaccharides, basing on the types of functions, these are of structural and storage type of polysaccharides, etc. But basing on the types of monomers or the types of monosaccharides that are present in a polysaccharide, this is of 2 types. You can write here, basing on types of units. There are 2 types of polysaccharides. One is homopolysaccharide. Another one is heteropolysaccharide. Homopolysaccharides are made up of same type of monosaccharide units. We can take here examples like starch, cellulose, inulin, chitin, etc. Under heteropolysaccharide, since when we are reading about plant anatomy, under parenchyma, we read as the cell wall of parenchyma has hemicellulose accumulations. There, hemicellulose is existed in a complete layer form, and there are many number of hemicelluloses which are forming into one cell wall. So we are calling there as a polysaccharide. But in case of hemicellulose, if you take one unit, then we are reading that as example under oligosaccharide. So here we will take hemicellulose as an example. Right, first meaning, it means made of many monosaccharide units, or not many, made of different monosaccharide units. Example you can take as hemicellulose. Now, basing on function, these homopolysaccharides are of two types. So, basing on function, homopolysaccharides are of two types. One is storage polysaccharide. Second one, structural polysaccharide. Under storage polysaccharide, we read the example starch, because starch is considered as a reserve food material in many types of plants. Example, potato if you take, in case of potato, the reserve food material is starch. Next, under structural polysaccharide, we can take example cellulose, because complete plant kingdom is having outermost cell wall made up of cellulose. Starch is made up of two parts. First part is amylose and second part is amylopectin. So starch is made up of two parts. What are those two parts? One is amylose. Second part is amylopectin. Amylose part of starch, it is made of 200 to 2000 monomers or monosaccharides. So, all the monomers are of glucose only. Means all the 200 or 2000 number of monosaccharides or monomers are of glucose only. Bond between these glucose molecules. Bond between glucose molecules or bond seen in amylose part of starch. Bond seen in amylose of starch is only one type of bond we can see, that is alpha 1,4 glycosidic bonds. Now, amylose is soluble in water. Amylose is soluble in water. Another point, amylose exists in helical form or in linear structure only. Amylose exists in linear chain form or helical form, means it can be existed like this, one straight line, or like this. It is not having any branches. Next, about amylopectin. Amylopectin is made of 2000 to 20,000 glucose units, means 1 unit of amylopectin. Glucose units or monosaccharides or monomers. Now, here the second point we have written, amylopectin also made up of glucose units only. So directly I have written, bond seen in amylose we have written here. But in case of amylase, bond seen in amylopectin, here we have to use bonds. Amylopectin R. In amylose, since there is only one type of bond, I have written like that. R. One is alpha 1,4 glycosidic bond. Another one branch is there, that is alpha 1,6 glycosidic bond. So in case of amylopectin, there are two types of bonds, alpha 1,4 glycosidic bond and alpha 1,6 glycosidic bond. For suppose alpha 1,4 glycoside bond is existed in such type of one chain, it will be having a branch. So here will be existed alpha 1,6 glycoside bond. So this is considered as the chain. So the contact between the chain and another linear form of glucose units will be showing 1,6-glycosidic bond. Now, this one, amylopectin, is not soluble or non-soluble in water. Non-soluble in water. But amylose is soluble in water. When we add iodine to starch, how to identify whether starch is existed in some types of substances or not? When we add iodine drops, then the color will be changed, of the substance will be changed to either dark blue or black in color. So, that dark blue or black color is obtained by amylose part of starch. We can write here, when iodine is added to starch, the dark blue or black color is due to amylose part of starch. So these are the points related to starch. Starch is made up of two parts. One is amylose and amylopectin. And amylose is soluble in water. Amylopectin is not soluble in water. When we add iodine to starch, the obtained dark blue or black color is due to amylose part of starch. Next example is cellulose. Cellulose is also a homopolysaccharide or structural polysaccharide of glucose. So, cellulose is a homopolysaccharide of glucose units. One unit of cellulose or one cellulose molecule is approximately made of 1500 glucose units. We have seen there in case of starch, amylose part of starch made up of 200 to 2000 glucose units. Amylopectin part made up of 2000 to 20,000 number of glucose units. In the same way, here one cellulose molecule, it is made up of approximately 1500 number of glucose units. Coming to the shape, there we have seen the bonds like alpha-1,4 glycosidic bond in case of starch. That already we have seen how the alpha-1,4 glycosidic bond is formed in disaccharide structures. In the same way in starch also it will be existed. Then what about cellulose? Is it cellulose is having the same type of alpha-1,4? Answer is no, because cellulose exhibits beta-1,4 glycosidic bond. So since all the molecules are pyranose forms of glucose, but one molecule of glucose in the cellulose molecule, it flips its direction or structure. Actually, it has to be like this. This has to be carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. Here also carbon 1, carbon 2, carbon 3, carbon 4, carbon 5 and carbon 6. But if the molecule shifts its position, for suppose, if the molecule is like this, it flips its shape upside down. Then how the oxygens or these carbons are changing their positions? Then if it flips, then oxygen comes down. The same carbon 1 of glucose, it will be of same. This is carbon 1, but this will be carbon 2, carbon 3, carbon 4, carbon 5, and here will be carbon 6. But in case of carbon 1 of one glucose molecule out of two which are side by side, here it will be OH will be on one side, and for C4 of the next or beside glucose molecule, OH will be here. OH. So the remaining carbon 2, carbon 3 hydroxyl groups are same for this one, and again this will be shifted. We can see oxygen atom here is on upside and here I have represented on the downside. So one glucose molecule of the side by side of any two which are side by side, it will be flipped, means the direction will be upside down. Now, there is a bond formed between by removal of again water molecule from these two hydroxyl and hydrogen of side by side glucose units. Then the bond formed will be represented like this. Means one side, alpha side, one side either right or left of one glucose is forming a bond with the OH groups or hydrogen of other side. Either again, like if we see this is the left side of the ring, and this is considered as the right side of the ring. In case of alpha-1,4, we have seen glycosidic bond formation between left side OH groups and hydrogens. But here we are seeing the bond is formed between left to right side. Now, such types of bonds are formed by dehydration, by loss of water molecule, zinc, and the bond is also called as glycosidic bond. But it is not of alpha, it is beta carbon 1, carbon 4 of the next decided upside down downturned glucose unit, beta 1,4 glycosidic bond. If you write a sentence form about this beta 1,4 glycosidic bond, we can write like flipping of one glucose unit of any side by side two glucose units causes beta 1,4 glycosidic bonds. Now, if you see what are uses of this beta 1,4 glycosidic bond, beta 1,4 glycosidic bond gives rigidity to the cell wall and also tensile strength to the cell wall. What is meant by tensile strength? Though the cellulose molecule is stretched, it will not get teared or broken down. Such strength of the cellulose is termed as tensile strength. So, uses of such beta bonds or beta 1,4 glycosidic bond. Use is giving rigidity to the cell wall, and providing tensile strength. Tensile strength means even after stretching there is no breakage. Actually, we use this term when we are explaining about water molecules in transport in plants chapter. This one term is existed in NCERT textbook also. But here we are just writing about what is use of this tensile strength. Helps from breakage even after stretching of cell wall. Uses. We'll write only few which are frequently asked in even maths, the following some sentence mode of questions, statement wise mode of questions. First use we'll write percentage of cellulose in cotton fiber. It is considered to be 90% of cellulose. Second one, percentage of cellulose in jute fiber or hemp fiber. These both belong to Fabaceae members. We used to write there as sun hemp. I think you might have seen this example in previous MCQs also. So these both belong to Fabaceae. It has only 40% of cellulose. Next, when cellulose is mixed with acetate, then it gives an artificial or some synthetic fibers which is considered to be as first man-made synthetic fiber, that is called as rayon. Cellulose acetate is used in rayon synthesis. This rayon is a fabric synthesis. Rayon fabric is considered as the first man-made artificial fabric. First man-made fabric. It is also considered as synthetic fabric. Next, fourth use of cellulose. Cellulose nitrate, when coated on plastic glasses or normal glasses, then it is called as shutter-proof glass. The glasses coated with cellulose nitrate are called or considered as shutter-proof, means there is very less tendency to break. Shutter-proof glasses. Next, carboxymethyl cellulose is a type of cellulose mixed substance that is used in many of the food items. Example, in case of candies, ice creams, this carboxymethylated cellulose is used as one of the compound. Carboxymethyl cellulose. This is indicated with capital letters, capital C, capital M and capital C. This is also called as cellulose gum. This is used in preparation of ice creams, candies, etc. These are the uses of cellulose. Next, if we see about glycogen. Glycogen is also an homopolysaccharide of glucose units. But in case of starch, we are seeing two parts, amylose and amylopectin. There in amylopectin, we are seeing two types of bonds. Same like that type of bonds in amylopectin. Glycogen also has alpha 1,4 glycosidic bonds and also alpha 1,6 glycosidic bonds. Now, glycogen is storage polysaccharide in liver and muscles. Liver and muscles. But in case of starch, when we add iodine drops, there we get dark blue or black color. That is because of amylase part of starch homopolysaccharide. But in case of glycogen, when we add iodine drops, we get red color. It gives red color with iodine. Next, if you see another homopolysaccharide, this is called the fourth, chitin. Chitin is again a homopolysaccharide of N-acetyl glucosamine. This one we represent as capital alphabets NAM. We see this part of NAM or N-acetyl glucosamine in peptidoglycan membrane of bacteria also. But there the combination is along with N-acetyl muramic acid in eubacteria. The glucosamine, N-acetyl glucosamine. This is nothing but representation of N-acetyl glucosamine, NAG, not NAM. I'm sorry for that. Okay. Now, this one we see along with N-acetyl muramic acid in eukaryotic peptidoglycan membrane. Now, chitin is present in, it is located in cell wall of fungus and in exoskeleton of arthropods like cockroaches and in exoskeleton of arthropods. Chitin is considered as second most abundant polysaccharide in nature. Abundant homopolysaccharide in nature. Then, what is the first abundant polysaccharide? It is nothing but cellulose. Cellulose is considered as the first abundant homopolysaccharide in nature. The second priority goes to chitin homopolysaccharide. Because of presence of only one type of carbohydrate, that is monosaccharide glucose units in the total chitin, we are considering this one under homopolysaccharide. Next is inulin. Inulin is again a homopolysaccharide of fructose molecules. Where is this inulin seen? Examples are in roots of dahlia plant. What is dahlia? It is one of the species that is related to chrysanthemum. Species related to chrysanthemum. Now, another polysaccharide is there, that is agar-agar. Agar-agar is a homopolysaccharide of galactose units. This is obtained from algae, means rhodophyceae members like Gelidium species, etc. It is considered as the solidifying agent. Is a solidifying agent. Next, seventh one we can consider as cellulose. Cellulose is also homopolysaccharide of glucose that is having different types of bonds, that is beta 1,3 glycosidic bonds. is a homopolysaccharide of glucose units. This cellulose has beta 1, 3 glycosidic bonds. Where it is located? It is located in the germinated pollen grain, in the pollen tube that is passing to get fertilized with embryo sac or egg of the embryo sac. Located in pollen tube. There, it prevents the backward movement of male to male gametes in pollen tube. Okay. Next, if you take some other examples, isabgol is one of the homopolysaccharide. Another one, hyaluronic acid, which is present in the synovial joints. Another homopolysaccharide. Pectin in the middle lamella. And anticoagulant, heparin, also considered as mucopolysaccharide, which are nothing but homopolysaccharides. Now, in the skin and in the cartilage tissues, there is one of the compound that keratin sulfate and chondroitin sulfates also. Those are also considered as homopolysaccharides, or simply polysaccharides we can call. So these are few examples that are related to polysaccharide, but which are very much important for your final examination point of view. I have written here. Already we have seen the heteropolysaccharide, that is hemicellulose. I told you when one hemicellulose unit that we are discussing, then we use a term like oligosaccharide, because one unit of hemicellulose is made up of 5 different types of monosaccharides. But when hemicellulose is being discussed in the cell wall form, in the parenchyma, outermost layer, etc., then we are considering hemicellulose total molecules in one layer as heteropolysaccharide, because it is made up of many monosaccharide units, not the single one. So for heteropolysaccharide, example is heteropolysaccharide or heteropolymer, we can call heteropolysaccharide. Example is hemicellulose. I already gave you what are those 5 different types. Cellulose, that is xylose, mannose, arabinose, galactose, and glucose. So since there are 5 different types of monosaccharides framing out into one unit, means one complete layer of the outermost parenchyma cells, we are considering that one as heteropolysaccharide.